So now we're going to start in on our population genetics unit, and here you really need to be drawing on your existing understanding of classical Mendelian genetics and the patterns of inheritance across individuals, and we're going to expand that now to look at what happens across whole populations of those individuals. And we're going to combine that with the concepts that we've already been talking about, those of allelic variation and natural selection, so that hopefully by the end of the unit we'll be able to answer a very specific question, and that is, how exactly does natural selection cause change in allele frequencies over generations? And of course you'll recognize this as our definition of evolution, allele frequencies over time. So the question is, how does natural selection cause evolution? In order to begin to answer that question, we need to reintroduce a topic or a concept that we've talked about a little bit, and that is the gene pool. And this is an illustration from your text, and what I really like about it is that it makes clear that when we're talking about the gene pool, we are not concerned about phenotypes. We're not even concerned about individuals. What we really care about here are gametes. And then what we want to know is what proportion of these gametes are carrying, in this example, the capital A allele, and what proportion are carrying the little a allele. We can calculate those two numbers or count them, and we call those the allele frequencies. The handy thing about allele frequencies, once you have those numbers, is that you can begin to ask questions about how and in what ratios may they be combined to form different genotypes. And anytime you're talking about combining alleles to create different genotypes, we can use a familiar concept, that of the Punnett square. Now here's a Punnett square that has been modified for a population genetics use. So just as in a traditional Punnett square, we have all of the gametes produced by one parent across the top, and all of the gametes produced by the other parent down here, down the left-hand side. And but rather than your traditional 2 by 2 column, 2 columns against 2 rows, we have changed the number of columns and the number of rows to match the allele frequencies of this population. So let's look at a concrete example. All right, so here let's imagine a gene pool where 60% of the gametes are carrying the big A allele and 40% of the gametes are carrying the little a allele. So then the allele frequencies for these two are 0.6 and 0.4 respectively. I can take that information and put it on my modified Punnett square by having 6 of these rows carrying the big A allele and 4 of these rows carrying the little a allele. And notice I put the same frequencies up here across the top because it doesn't matter if we're talking about the father or the mother. I can then just use it as I would a traditional Punnett square. So I would combine the A here and the A here to create my homozygous dominant genotype. I would combine the A from here with a little a from here to create my heterozygous genotype. I could take an A from here and an A from here to create my other group of heterozygotes, and then an A from here and an A from here to create my homozygotes. Now in this illustration, we've given a concrete number of zygotes. So this is only 100 zygotes. But drawing this out is fairly cumbersome, and certainly if I had 650 zygotes, it would be even more of a pain. So I need a simpler way to do this, and so I can just do it using probabilities. So here I have the same information, but I'm just calculating it arithmetically. So I have the probability that an egg will contain the capital A allele, which is equal to the capital A allele's frequency in the gene pool, and I have the probability that the sperm will contain the capital A allele, which is right here, and then the probability that these two cells will fuse to form the zygote is the probability of this times the probability of that, which gives me the probability of a homozygous capital A zygote of 0.36, or a 36% chance that this gene pool will give rise to a homozygous dominant zygote. Again, down at the other end, I've got the probability that the egg will carry the little a allele of 0.4, the probability that the sperm will carry the little a allele of 0.4, and then the probability that they'll combine, 0.4 times 0.4 is 0.16, or a 16% chance that this gene pool will give rise to a homozygous recessive zygote. For the heterozygotes, I just need to make sure that I understand that the probability that an capital A little a heterozygote will occur is 0.24, and the probability that a little a capital A heterozygote will occur is also 0.24. And more importantly, that these two cells are entirely equivalent. There is no difference. So I can add these two probabilities together for the total probability of a heterozygote of 0.48, or a 48% chance that this gene pool will give rise to a heterozygote. Notice the advantage of the probability method is that I have fractions. So now this 0.36 number gives rise to 36 individuals if I have 100 zygotes, but I could use the same value to calculate the number of homozygous dominant in a population of 650 or 3,832, right? So I'm not dependent on an absolute number. I've just got ratios, fractions of the relationships between them. Now, it's also important to understand that it does not matter if a population has a gene allele frequency of 0.6 and 0.4, or if the population has an allele frequency of 0.8 or 0.2 or 0.1 and 0.9. Right? I need a generic case that can show me the relationship between the allele frequencies and the genotype frequencies. And so for that, I turn to Hardy-Weinberg. Right, so here's Hardy-Weinberg, and we'll just walk through a quick example where we have these three genotypes. So we've got homozygous A1, homozygous A2, and the heterozygote A1, A2. And if I want to know something about the genotypes here, I might want to know about the frequencies of the genotype. So I want to know the percentage of the population that is homozygous A1, or the percentage of the population that is homozygous A2. So to calculate those, I would just count up all of the homozygous A1 individuals and divide that by the total number of individuals, and that would give me a genotype frequency. Now that's cumbersome to go around counting everybody, and Hardy-Weinberg came up with these generic terms for the frequency. So here Hardy-Weinberg says that the frequency, or the percentage of the population that is homozygous dominant, is represented by this term p squared. The percentage of the population that is homozygous A2 is represented by this term q squared. And the percentage of the population that is heterozygote is represented by this term 2pq. Further, they said that if we add the percentage that is homozygous dominant to the percentage that is heterozygote to the percentage that is homozygous recessive, if I add all of these three terms together, then I will have counted all of the individuals in the population, and so the percentages should equal 1. Okay, so I can depict this graphically with the familiar sort of Punnett square nomenclature here, or imagery. So this large yellow square represents all of my homozygous dominant genotypes. These are all of the individuals with the heterozygous genotypes, and these are all the individuals with the homozygous recessive genotypes. And if I add all of those individuals together, then I've counted all of the individuals in the population. Now further, I would want to know something about the gametes that these adults would make. So it should make sense that all of these adults will make gametes carrying the A1 allele. All of these adults will make gametes carrying the A2 allele, and that half of these gametes will be carrying the A1 allele, and half of these gametes will be carrying the A2 allele. And if I add up all of these alleles plus all of these alleles, I'll have the total number of alleles. Now I can take that total number and divide each of these by that total for a percentage or a frequency. And so Hardy-Weinberg came up with these generic allele frequency terms and said, if I take the genotype frequency of the homozygous plus half of the genotype frequency of the heterozygotes, I get this term P, which is the frequency of the A1 allele in the subsequent gene pool. And if I take half of the genotype frequency for the heterozygotes plus all of the genotype frequency for the A2 alleles, I get this term Q, which gives me the frequency of A2 in the next gene pool. And that if I add the frequency of the A1 allele to the frequency of the A2 allele, I should have all of the alleles in my population, and that should equal 1. So when you're studying Hardy-Weinberg, it's important to have these P's and Q's straight. So just as a quick reminder, P squared, 2PQ, Q squared, these are genotype frequencies, the proportion of the population that is homozygous or heterozygous. P and Q, on the other hand, are allele frequencies, the proportion of the gene pool that is carrying the A allele, A1 allele, or the A2 allele. Now, in these examples, here we used the allele frequency to calculate the genotype frequencies. And then in this example, we used the genotype frequencies to calculate the allele frequencies. And as long as you can do this, as long as you can go from allele frequency to genotype frequency or genotype frequency to allele frequency, then we say that the population isn't changing. The allele frequencies will stay the same from generation to generation. The population is not changing. So Hardy-Weinberg called this the equilibrium, and they more formally defined it as by saying that the population is in equilibrium if these two conditions are met. One, that the allele frequencies do not change from generation to generation, and two, that the allele frequencies can be used to predict the genotype frequencies. So Hardy-Weinberg is what we call a null model, because as long as Hardy-Weinberg is in equilibrium, then the allele frequencies are not changing. If either of these two conditions or conclusions are not met, then it means that they are changing, and that means that they are evolving. Okay, so next we'll look at some forces, some mechanisms of evolutionary change that can cause populations to deviate from equilibrium. That's in our next video.